Okay, so welcome to our lecture on viruses and viral genomes, and we'll give you some clinical examples spread throughout this lesson as well. So first off, you have to ask yourself, what exactly is a virus? Okay, so when you hear virus, I want you to think infectious packages of genetic info. And I'm going to keep emphasizing throughout this lecture, whenever you hear virus, I want you to remember that a virus is mainly two parts put together, okay? The two parts that will be put together is a nucleic acid, which can either be RNA or DNA, within a protein capsid shell, okay? So you have nucleic acid within a protein shell, and that's going to keep coming up throughout today's lecture because of the idea that since a virus is just nucleic acid and protein shell, that will guide a lot of what they're doing. Their ultimate goal is to make more nucleic acid, make more proteins, and put together more viruses. Okay? We'll come back to that a little later on. Now, the next point I want to make when it comes to viruses is that they are not considered self-sufficient living cells. So you have to ask yourself, why? Why are they not considered true living cells? Well, in order to qualify as a true living cell, you have to be able to do the central dogma on your own. And if you remember, the central dogma is DNA to RNA to protein, which means you have to be able to create RNA sequences from genome, from DNA, and to synthesize proteins through having your own ribosomes and being able to generate your own energy. But we just said viruses, all they have is nucleic acid in a protein shell. So they don't have their own machinery like ribosomes to undergo the proper central dogma. Okay, they need to rely on a host cell in order to make proper mRNA and then proteins. Okay, so they basically store their genetic messages, but then they relay that onto the host cell to make mRNA. So what we call them is obligate intracellular parasites. So make sure you write down that viruses are obligate intracellular parasites. They are required to go inside host cells, and they feed off of that host cell's resources, okay, in order to be able to survive, okay? So they're not truly self-sufficient living cells. They cannot do, things like replication without a host cell, okay? They cannot do the central dogma on their own. Now, when we say that viruses have their nucleic acid, that's their genome. And that means that a viral genome can either be DNA or RNA. It can be single-stranded or double-stranded. And sometimes you encounter circular versus linear genomes. Okay, so you're going to see throughout this lesson that whenever we talk about viruses, a lot of times you'll hear people refer to what type of genome they have, and that can kind of affect how they interact and what they do. But like I said, we're going to go into more detail with that after. Just be aware that viral genomes vary. Okay, they can be DNA or RNA. So now, when we talk about a virus's genome, like I said, when it comes to viruses, they are nucleic acid and a protein shell, which means that when we ask ourselves, what proteins must the virus make? Well, their two ultimate goals is to replicate or make more of their genome and make more of the protein shell to assemble new viruses. Now keep that in mind. I'm going to keep saying assemble throughout this lecture because Viruses don't reproduce, they don't divide, they don't have sexual replication. They assemble new viruses. They make more genome, more protein, and put them together. Okay? So the proteins that they have to make are proteins or enzymes involved in replicating their genome, and proteins or enzymes involved in making more of the protein coat and assembling new viruses. So in order to do that, they have what's called early versus late genes. Their early genes are what will code for the enzymes needed to make more viral genomes. Okay? So early genes code for enzymes for nucleic acid replication. So for instance, things like a polymerase. Okay, so early genes are for nucleic acid replication, whereas late genes are for making the protein coat, which means it's for viral assembly, putting together new viruses. Okay, now in addition to knowing early, the nucleic acid replication genes, versus late, the protein coat genes, you should also be aware that the viral genome has what's called monocistronic viral mRNA. That is a fancy way of saying that they produce one long piece of mRNA that then codes for multiple proteins. So when you look at this figure here, if you were picturing this long monocistronic nucleic acid, you end up ultimately from that one long sequence, you produce one long protein that then gets chopped up into the individual proper little proteins. So monocistronic means you have a string of multiple genes put together, and then ultimately they produce one large protein that gets chopped up into the individual usable little proteins. Now, this slide is just to show you that there's a whole bunch of different categories of viruses. Okay, so I could be mean and say that you have to memorize every single one of these. Of course, I'm just messing with you. I just want you to see the different types of categories that we have for viruses. So notice in this case, a lot of viruses can be categorized based on their genome. So all of these are categorized as having double-stranded DNA versus single-stranded DNA, and then so on and so forth. Sometimes they also categorize viruses based on their shapes. Okay, so is it a helical virus or or a complex virus, something like a bacteriophage. Okay, so just know that viruses are usually categorized based on their genome or on their shape, which can also be called their structure. Okay, now we're going to go one by one through some of the viral genomes. This first slide is to show you viruses that have DNA genomes. Okay, these are DNA viruses that can infect animals, and the examples I want you to know are simian virus, herpes virus, and pox viruses. Now, when you hear any of these three names, I want you to be able to recognize, oh, that has a DNA genome. Okay, so if I said what type of genome does the herpes virus have or pox viruses, you should know right away, DNA genome. Okay. In addition to knowing what type of genome they have, I want you to know the main significance or examples of each of these viruses. So for simian viruses, when you hear the word simian in the English language, you have to ask yourself, what does that mean? Well, simian means monkey. So simian virus actually causes cancer in monkeys. Okay? Then we have the next one, herpes virus, which everyone always hears of, but they automatically only think of the STD herpes. They don't realize that herpes virus is actually a category of viruses, okay? There are 8 herpes viruses, okay? And if you think about the STD herpes, that will kind of help you gain a little insight about the 8 different types of herpes viruses. So when you think of herpes, the STD, one of the things that automatically students think of is, boom, you have herpes for life, okay? That's very difficult to cure because it goes latent at various stages, okay? And usually these viruses go latent in nerve cells. So when they reactivate, it's very, very painful. And one of the things we're going to mention a few times throughout this course is when exactly do latent viruses tend to reactivate? And we're going to mention that in other slides as well. Always keep in mind, reactivation tends to be when someone is either immunocompromised or if they've been under a lot of stress, okay? But now getting back to the herpes viruses, okay? Automatically you think of the STD herpes, so that's type 1 and type 2. Think about other viruses that go latent, and that will help you think of the other herpes viruses, which include mono, which is the Epstein-Barr virus, and chickenpox. Chickenpox is actually a herpes virus. I want you to write that down, put stars, highlight it, because most people assume that chickenpox is a pox virus. It is not. The only reason it's named chickenpox is because you get a pox on your skin, which is those little speckling, those dots that you get all over. Okay? The actual virus itself, though, is a herpes virus. And then what does chickenpox eventually become after it's been latent in your body a while? Shingles. So shingles is also an example of an infection from herpes virus. Okay, so make sure you remember herpes viruses tend to go latent, so they're for life, and that chickenpox and mono are actually examples of herpes viruses, not just the STD herpes. Now, the last category we have here is pox viruses. So remember, chickenpox is not a pox virus, it's herpes. The pox viruses are actually the largest animal viruses. And a main example of a pox virus is smallpox, okay? We're going to see that a little more in another lecture, right? So again, keep in mind that anytime you hear simian viruses, herpes viruses, pox viruses, or any of the examples I just gave you. So for instance, if I asked you about chickenpox, you should know right away that it's a herpes virus, which means it has a DNA genome, okay? Make sure you know that any of these 3 viruses and their examples have a DNA genome. Here is some recap information so you can pause the video and write down these notes. Now, this is to show you how animal viruses, which include simian viruses and herpes viruses, can induce cancer. So for instance, herpes viruses, we just said, include things like mono, okay, Epstein-Barr virus, and they have recently found that not only does mono stick with you for life and you end up getting chronic fatigue syndrome, but it also down the line can cause cancer, especially nasopharyngeal cancer. Okay, so you have to be careful with any of these infections because, you know, it's not like, oh, I could just pop a pill and suddenly I'll feel better. No, a lot of these viruses stay with you for life and can cause cancers. So this slide here, this figure over here, demonstrates how viral DNA can end up leading to tumor cells in a patient. So when you look at this, I know it can look a little scary or confusing at first, but this figure is basically just showing you that viral DNA, so that would be this blue ring up here, viral DNA is able to cut itself open, cut open host DNA so that it then glues itself in, okay? Now it uses host resources that will be replicating and expressing the host genome to also replicate and express its own DNA. Now, within its DNA tends to be genes or cells that will basically trigger hormones that cause uncontrolled cell proliferation. Okay, so basically the viral genes will cause uncontrolled cell proliferation, so there's no off switch in the host cell's cell cycle. So if you think about it, uncontrolled cell proliferation is the definition of cancer. Okay, so these genes will encourage hormones or products that will just cause your cells to keep replicating, and clumping up into these big tumor cells. Okay, so again, this is something we'll mention various times throughout the course as well. Always keep in mind that viruses and infections have this ability to cause cancer. Now, in addition to there being DNA viruses, there are also RNA viruses. And when we talk about the RNA viruses, there are a few different types. First off, I want to note that the RNA viruses have the smallest genomes. They have very high mutation rates, okay? So even though they're small genomes, they have very high mutation rates, and that means that they're great at evading the host. And it also means that it's very difficult to produce vaccines sometimes for certain ones if they're replicating too fast and having too many mutations. Okay, so a lot of them we have now developed vaccines, but it takes quite a bit of effort for the RNA viruses. Now, there are three main varieties that you should be aware of. There's double-stranded RNA genomes, positive single-stranded genomes, and negative single-stranded RNA genomes. Now, what I want you to put stars next to are the positive single-stranded and the negative single-stranded genomes. Okay? When you see positive, I want you to think positive, think protein. Okay? Because the positive single-stranded RNA is actually, it matches the mRNA that would make proteins, okay? So it's the proper code to make proteins, whereas the negative strand is the complement to the proper strand for protein making, okay? So a negative strand will not properly make proteins. It has to do extra steps of making positive strands or its complement before it can actually make proteins. Okay, so I'm going to write that down in a summary slide on the next slide so that you can have it in your notes, because sometimes that's a little confusing to students. And then we're going to go through each of those types just to really solidify that point. Okay, so this is the recap slide. You can pause it if you need a minute to write down what I was just saying. The reason I wrote it out is even though we're going to say it a bunch more times in the next few slides, I want to make sure that you're very comfortable with this idea of positive, think protein, okay? So here we have the positive stranded RNA viruses. Okay, their examples include polio, the common cold, Hep A, and as you can see, like we mentioned earlier, these are monocistronic, meaning that they will make a polyprotein, which is a really, really long protein that, as you can see in this diagram, then gets chopped up into its individual little proteins. Okay, now when you look at this, you have to what is the significance of plus RNA, okay, which we also call sense? Now, anytime you see plus RNA, or sometimes we call it positive stranded RNA, sense RNA, no matter which of those terms you see, you have to tell yourself, hey, positive stranded viruses are lucky. They're lucky because they have their mRNA code already. They can make more protein. Now, as we kept saying, viruses only need to do two things. They need to make more of their genome, and they need to make proteins to assemble new viruses. The positive RNA guy, he can do it all already. By making more of his positive strands, he makes more of his genome, and that positive strand can be used to make protein. Okay, so that's very significant. Okay, when you look at this figure, you can see this area here is for making protein, and this area here is for making more of the nucleic acid. Okay, so he can take care of everything. Negative stranded RNA viruses, on the other hand, they have a little more work. Okay, they're called antisense because they're the complement of what they need. Okay, examples are rabies, mumps, measles, influenza, and Ebola, all things that I'm sure you've heard about. And you have to ask yourself, just like we did with the positive stranded RNA, ask yourself, what's the significance when you hear minus RNA or antisense RNA? Why does the negative stranded virus end up not being as lucky as the positive? And so what's the problem here? Well, its genome is minus RNA. So it can make more copies of that, and yay, it's done its genome job. But if it wants to make proteins, what's currently the problem? It needs positive strands. Okay, positive is for protein. So the negative strand now has to make copies of its complement. Okay, it cannot get by with just negative stranded RNA. Okay, so it has to become like a little factory, which you see in the figure on this slide, to make not only more of its genome, the negative strand, but also to make its complement, the positive strand, which makes proteins. Okay, so you can see in this slide here, it makes negative for its genome and then positive to make proteins. Okay, so it's got extra work. Okay, if you have any trouble with that idea of positive versus negative, just send me a remind message and I'll go over it in a little more detail. Now, another category of viruses or another genome are the retroviruses, such as HIV. Now, with retroviruses, they have single-stranded RNA, and then they have a double shell around them and an extra outer envelope. Okay, so they're a little more complex. And what's weird about them is that rather than using their RNA to right away make proteins, these guys use RNA. But they then, once they're in a host, they make DNA copies from that. Okay? So what they do is they have a series of enzymes, which include reverse transcriptase, integrase, and protease, so that what they can end up doing is they take their RNA sequence. Once it's in a host, they make a DNA copy of it using reverse transcriptase, and then they use integrase to get that DNA copy into the host DNA. And so now the host, just like we showed in earlier slides, the host now has viral genes in their DNA. And whenever they go to replicate and express their own genes, well, now they're doing that work with viral genes as well. Okay? Now, in order to do this process, they also have what's called long terminal repeats. Okay, so within their DNA copy, they have these sequences that get cut that allow for the, basically gluing in of these viral genes into the DNA host. Okay, now what I want you to take out of this is that these guys, they're going into the host DNA. So that means they're there to stay. Okay, so the retroviruses you're going to have for life because now they've gone into your genome. The other point that I want to make is that they use reverse transcriptase to make their DNA copy, and reverse transcriptase is ridiculously bad at its job. Okay, it makes a ton of mistakes, and I'll refrain from comparing it to any of my students, but you know who you are. There are just these enzymes that make these tons of mistakes, and mistakes in DNA, what do we call that? Mutations, right? So retroviruses are going to make a lot of mutations each time their genes get replicated. And what's important about that is that makes these guys even more difficult to treat or to handle. Okay, so not only are they there to stay because they're integrating into your genome, but they're also problematic because they are constantly changing through all of these mutations because reverse transcriptase is so bad at its job. Okay, so make sure you're comfortable with the idea of reverse transcriptase causes mutations. Write that down. Reverse transcriptase causes mutations, and it doesn't have any proofreading to correct those mutations. Okay? Okay, so here we have the retrovirus genome, which is rather unique, and it contains 3 specific genes that I want you to be aware of, which we show right down here. Okay, so that's GAG, POL, and N. GAG, or GAG, what that's coding for are the structural proteins, which will make up that outer protein shell, which we call the capsid. The POL, similar to what you've seen in previous lectures, POL encodes for reverse transcriptase and integrase. Okay, so these, kind of like when you think POL like polymerases, these will be involved in the genomic replication and integration. Okay, keep in mind again, reverse transcriptase, very bad at its job, leads to a lot of mutations with no proofreading. Okay, again, kind of like some students I know. And then you have the ENV gene, which, like it kind of hints at, is for the envelope proteins, meaning that the ENV gene will code for the proteins that make up that extra outer membrane that we said retroviruses have. Now, when you talk about retroviruses, there's a main process that they go through. The first thing that they have to do is enter a cell, and then once they enter a cell, they have to remove their virus envelope, okay, because remember they have that extra envelope, and they leave that behind at the cell's membrane. They then have to worry about their genome, right? Because big thing, if you're a virus, you want more genome, more proteins. So what they do is reverse transcription of their RNA genome, okay? Meaning they're now going from RNA genome to make a DNA copy, reverse of transcription, right? After that, now that they have a DNA copy, they have to integrate that viral DNA into the host DNA, the host genome, right? Once it's there integrated in, they need transcription to occur, meaning expressing those viral genes. Once they've been expressed and they have the proteins they need, then they have assembly and packaging of the newly made genes. And then they ultimately have budding of the enveloped viruses releasing from those host cells. And the reason why after that they now have an envelope around them is because if you look at this picture here, they're stealing part of their host membrane. So that extra envelope or membrane around retroviruses, anytime you see a virus with an extra envelope or membrane, just know that they're jerks and they totally stole that from their host cell. Okay? I'm going to recap these steps because I know it was a lot of information on the next slide. Okay, so here's just a recap of those steps that I just went through. So you can pause and write them down. And again, if you have any questions or you're uncertain of anything, just send me a remind message. Now this slide is just to help you get a closer look at what we mean when we keep saying reverse transcription and the retrovirus synthesizing DNA. Okay, so if you look at these figures, here we have regular transcription. You're going from double-stranded DNA, where one of those strands, the antisense strand, serves as the template to make the RNA strand. In reverse transcription, however, the retrovirus has an RNA strand as its genome. What it does is it uses reverse transcriptase. Again, we said very bad at its job, makes mistakes. It uses that to make a single strand DNA copy. Once it has that, now it can degrade or get rid of its RNA strand and then make a second copy, the complement to its single DNA strand, so that it has proper double-stranded DNA, which it can then cut into the host DNA. So it's taken its double-stranded DNA copy and it's integrated it or inserted it into the host genome. So now when the host goes to replicate or express its own DNA, boom, it's now replicated and expressed viral genes, and it's ultimately made viral RNA. Okay? So again, keep in mind in this process, reverse transcriptase, very bad at its job, makes a lot of mutations or mistakes. Okay? Please keep that in mind whenever you hear reverse transcription or retroviruses. Okay? Now we get to the idea of infecting, okay, how these viruses are infecting their host. A lot of viruses, what they'll do is they will code for special enzymes that are critical to infection. Okay, so two big viral enzymes are lysozyme and neuraminidase. And the students make fun of me because I mispronounce it every time or I stutter when I try and say it. Okay, so neuraminidase and lysozyme. What these do, first you have lysozyme. And you've probably heard that in terms of our own cells as well. Lysozymes can make holes in cell walls, okay? So they're able to lyse the cells that they are trying to infect, okay? So you especially see these two enzymes in viruses that infect bacteria, okay? Viruses that infect bacteria tend to use these enzymes. So they use lysozyme to make holes in the cell wall of the bacterial cells. And then they use neuraminidase. Again, I hate trying to say that word. What those do is cleave the glycosidic bonds and basically allow the liberation of viruses from cells. And when you hear the word glycosidic, you're like, oh, what does that mean? That sounds like a weird word. Well, you've heard a word that's very similar. Peptidoglycan, okay? So these, again, these kinds of bonds are usually found in the cell walls of bacteria. So that's why I said that these, the viruses that contain these two enzymes will usually be infecting bacterial cells, and the overall purpose is to try and break the bacterial cell wall to basically get in or get out of that bacterial cell. Okay, so I'm going to have the next slide have the definitions written out just so that you know that you had it written properly. Okay? Okay, so here's the recap in case you were unsure of any spelling or exactly what I was saying for those definitions. Now this slide gives you an overview of the virus life cycle as it's infecting bacterial cells, because again right now we're mainly focusing on bacterial cells. The first step, as you see, is attachment, which is kind of like when you think about any infection, even in us, in order to infect a host, the pathogen needs to be able to stick to it first. So to grab a hold, stick to it, and then to enter, okay? Now, entry of the virion is something that will occur if the virus is infecting an animal cell. So the whole virus will enter. But if it's infecting a bacteria like the figure on this slide, instead of the whole virion entering the host cell, only the nucleic acid will be dropped in. Okay? Once the virus particle or its nucleic acid is inside the host cell, well, then it has to do what we've been saying over and over throughout this lecture, which is make more copies of its nucleic acid and make more copies of its protein. Once it's made a whole bunch of copies, which you see happening here, it then gets to assemble or put together new viruses. Because remember, viruses do not reproduce, they do not divide, okay? Viruses assemble. Make sure you write that in your notes in bold and highlight. Viruses assemble. They do not reproduce or divide. Once they have assembled new viruses, then that's when they burst out or release from their host cell. So again, the main goal, what they got to do, grab a hold of a host cell, get inside of it, make copies of their genome and protein, put together new viruses, and then pop out of that cell. Now, when we talk about viruses, what's interesting is you have what's called a one-step growth curve. Okay, whenever you think of viral growth curves, I want you to think of one-step growth curve. The reason it's called that is that if you were to do a time course study looking at, a viral infection in culture, okay, so picture, growing a bunch of media, having bacterial cells in there that you're infecting with viruses. You won't see any increase in virus number until, boom, those viruses start bursting out of the cell. And when they lyse out of a host cell, now you have a whole bunch of new viruses released at once. Okay, so all of that stuff we saw in the previous slide, such as them replicating and assembling, you can't see that until they burst out of their host cells. Okay, so when you're doing a study trying to look at the viral amount in a culture, you'll basically see nothing, no change, and then boom, a big jump in the number of viruses present because now they have burst out. Okay, so basically when you look at a one-step growth curve for viral replication, you see a latent phase, which is the flat line. You're not detecting any new viruses because at that time they're basically, replicating their nucleic acids, they're making more protein coats, and they're starting to assemble new viruses. But it's not until the burst or the release that you suddenly detect in the culture the big jump in viral number, okay? Because before that, in the latent period, they're still inside of their host cells, okay? So you cannot see an increase in their amount, okay? So whenever you think of whenever you think of a viral growth curve, think one step. You do not see the jump in viruses until they burst out, okay? And this curve here, that is the burst, okay? Now, when we talk about actually detecting the viruses and counting, the concentration of the viruses, which we call the titer, what you can do is the plaque assay, and we're actually, we do that in the lab, okay? So you'll get a chance to actually do this hands-on. Now, there's some terminology on here that I want you to be able to define. First off, the term titer, T-I-T-E-R. What that means is the number of infectious units per volume fluid. okay? A fancy way of saying how many viruses you have in that culture, okay, infecting the host. To do this, you do a plaque assay, which is basically a plaque is an analogy to the bacterial colony. Okay, so rather than on those agar plates that we work in the lab, rather than seeing a clump of beige bacteria, instead a viral plaque is an empty space, a little circle in the growth. It almost looks like a very small zone of inhibition. Okay, the reason you see these viral plaques, okay, is because there was bacteria there. The virus infected the bacteria, and what do viruses do to bacteria? Boom! They cause them to lyse, right? So those little holes that you see represent where bacteria burst or where bacteria lysed because it got infected by viruses. Okay, so that helps you to kind of get an idea of how much of the virus is present. Now notice that there is a formula on the bottom of how exactly you do the plaque assay, okay? And on the next slide, I'm going to demonstrate and, have it written out, an example of using that formula, trying to see exactly how much virus you have infecting the bacteria in your sample. Notice the unit will be PFU per ml. That stands for plaque forming units per ml, which is similar to in microbiology and general micro, if you remember the term CFU per ml, which was colony forming units per ml that we talk about with bacteria. Okay, so I know this sounds scary and a lot when you hear it in words, but I'm going to use a written out calculation example to kind of help it become a little clearer in the next slide. Okay. But just for now on this slide, know if you want to estimate the amount of virus infecting bacterial culture, you can perform a plaque assay. Okay, so here's the example of using a plaque assay. Okay, if you use 1 ml of virus stock in 9 ml of water, meaning you diluted the virus, okay, and whenever you calculate a dilution like this, you do the amount, which is 1 ml, over the total, which is 10 ml. Okay, so you diluted it 1 over 10, and then you plated 0.1 ml, and you got 160 plaques. So the way that you would calculate the viral titer, or the viral load, is you would use the formula on the previous slide, and you would do 160 plaques over the total dilution, which is the 0.1 ml plated times the 1 over 10 that you diluted that sample. Okay, and then the final unit would be PFU per ml. If you're not sure how we plug that in, or you want extra practice, just send me a remind message asking me for more practice problems of viral titer. Okay? Now, when we talk about viral infections, there are two terms that you'll frequently hear, which are lytic versus lysogenic. Okay? In lytic infections, the virus, if you think about lytic, that means lysing, bursting. So you know that the virus is going to destroy the host cell. How would they do that? Well, they'd be replicating their genome, but also producing proteins and making more viruses. Okay? So when we say what occurs in lytic, that's not occurring in lysogenic, well, first let's take a look at lysogenic. Lysogenic is considered dormant, right? They're not lysing the host cells. They're not breaking free, which means that they're not making new viruses, right? Because it's only when they make new viruses that they're going to burst out of the host cells. So when I ask what occurs in lytic that does not occur in lysogenic, protein synthesis, right? So in the case of lytic, you have the virus replicating its genome and producing proteins, assembling new viruses. Whereas in the lysogenic, when I ask what is the only thing the virus is doing, it's only replicating its genome, which you see in this figure here. Okay, it is only replicating its genome. It is dormant. You can't tell it's there because it is not producing proteins, not making new viruses. And viruses switch back and forth between lytic and lysogenic frequently. So now we'll go through different types of infections. Okay, so first you have infecting prokaryotes. Okay, so these are going to be the viruses that are infecting bacteria, for instance. First one we have here, you have bacteriophages here. Okay, as the name suggests, these are viruses that infect bacteria. Now, notice if they're going to infect bacteria, they look kind of like syringes because they have to be able to inject the nucleic acid into the bacteria because bacteria have that tough cell wall that prevents them, the virus, from just merging with the membrane the way that they would with animal cells. Okay? Now, in order to infect the bacterial cells, you have the need of that virus to first be able to attach. And notice, even like with our cells, anytime a virus is going to attach, it has to be highly specific, okay? And so the big way that viruses attach and enter, ultimately, cells is through basically receptors. They'll have complementary receptors to the surface of the host cell. This is important to know because that means that a big way to target viruses is to interfere with receptor recognition. And in fact, the people who have HIV resistance is because they have mutations in the receptors for our host cells. Okay, so just like with our host cells, viruses that want to infect bacterial host cells depend on receptors. Now that's all fine and dandy, but we don't really care when the viruses are infecting bacteria, right? We care about when viruses are infecting animal cells. So here you have an overview of animal viruses. Notice that when the virus was infecting bacteria, they were just injecting their nucleic acid in. With infections of animal cells, the whole virus enters, meaning the nucleic acid and the protein shell. Okay, so make sure you put stars next to that difference. Okay, now keep in mind that with animal viruses, you tend to see a lot of enveloped viruses, right? Why is that? Because a lot of them are stealing that envelope, that membrane from the animal viruses. Okay, so as I mentioned before, any virus that has an extra membrane on top of its protein shell, where did they get that? They stole it. They stole it from their previous host cell. So if they're infecting your body, they'll go to one of your cells. When they go to burst out of it or to bud out of it, they will take part of the membrane. Why is that valuable to them? Well, that membrane is yours. It has your receptors on them. And that allows them to do two things. It allows them to merge with your other cells to infect them because it's the matching receptors. And it helps them get around your body without your immune system being able to target them as well because they look like your cells. Okay, so it's important to know that viruses steal membranes from the host. Now, there are a few different things that can happen when a virus tries to infect your cells. Okay, we call them the various consequences of viral infection. The first one is what we call a failed infection. that means the infection was abortive. The virus did not successfully infect. The second one is a lytic infection. And anytime you hear lytic, well, you know what that means. It means cells are lysed, host cells die. The infection can also be persistent. Okay, what persistent means is you have replication and release of the viruses, but without the cell dying. Okay, so in lysis, the cell dies, the host cell dies. In a persistent infection, the viruses are budding out of the host cell, but they are not lysing that host cell. So the infection can persist in that cell. You then have latent recurrent infections. What that is you don't have virus production because it's latent, they're dormant. But it has the potential for recurrence, meaning that the latent infection, as you can see here, can revert to lytic. Okay, so meaning a dormant latent infection can become active and start killing host cells. The last one is viral transformation, and that consequence is not like the bacterial transformation that we talk about in lab. Viral transformation, that's seen here. That means the virus has triggered tumor cell production. Okay, so viral transformation is causing cancer in the host because, as you saw in the previous figure, sometimes the viral genes include oncogenes that once they're integrated into the host genome, now that host ends up producing the hormones or products that ultimately lead to uncontrolled cell proliferation, cell replicate, boom, cancer. Okay, so I'm going to summarize each of these on the next slide just in case you had trouble writing down any of the definitions. Okay, so here are the recap of the possible outcomes for you to write down in your notes. You can pause the video here. Then we have the basic steps in viral disease as it's basically going in the body or progressing in the body. Now, rather than kind of discussing through this on this slide, I'm going to actually skip to the next slide where I will recap it in the notes for you to write as I go along, because it can be kind of overwhelming if I'm just speaking the 8 steps. Okay, so here I wrote out the 8 steps, which I will not make you memorize for the exam, but just to go through, the first step you have here is the acquisition of the virus and the infection. Then it will initiate at a primary site, so that's the first site of infection. And it will activate innate protections. What that means is your innate immune system, the first line of defense that your body has, basically things like barriers or enzymes. You have tears are part of your innate immune system. Saliva is part of your innate immune system. Even things like your skin and sweating and hairs throughout your body, that's all innate immunity, as well as general macrophages that go around trying to engulf various pathogens. Also, part of the innate protection when it comes to viruses is various proteins that your body will release. Now, it then goes into incubation stage, and that's when it can start to spread to other sites, and you start to get replication in your target tissues where you start to really see the symptoms of infection. Then you get the host responses, and that is basically now, rather than just the innate immunity, now you have the specific adaptive immunity. Okay, your body is really fighting this infection, and you then end up with virus production in tissues that could then transmit to other people. And then lastly, you end up either by getting better, your immune system did what it should and protected you, or you end up with persistent chronic infection. So that was the one in previous slides where you saw the virus stays in your host cells replicating, and it releases a little bit at a time without killing those cells. Okay, like I said, you don't have to memorize these steps, but just be aware there are 8 stages that the viral infection will go through in your body. Now, when we talk about actually getting these kinds of infections, there are various factors that will basically determine how easy it is for you to get infected by a virus and how bad the symptoms will be once you get infected. Now, things like mechanism of exposure and site of infection matter, okay? Mechanism of exposure and site kind of means, well, how did you pick up this infection? Okay, was it through a big gaping wound in a surgery? Was it through inhaling just a few particles, a few viral particles, stuff like that? The second bullet point is very, very important. Put stars next to that one. Your immune status, age, and general health. What that means is once the host is infected, the host's immune status and immunocompetence are major factors in whether or not that viral infection, or any infection really, will be life-threatening, benign, or show no symptoms at all. Okay? So think about it. If someone is very, very young, so a baby, or very, very old, the elderly, and if they're currently immunocompromised, they already have comorbidities, they're more likely to end up having life-threatening symptoms from an infection. Whereas that same infection, if it's just in, a healthy, active, fit, mid-age or 20s, 30s type of person who's immunocompetent, that person may barely sniffle or sneeze, okay? So your immunocompetence is very, very important, or if you're immunocompromised. Also, the genetics of the virus and the host. So for instance, when we went through the slides, you saw certain genomes are tougher than others, such as retroviruses like HIV has. The last one is viral dose. Okay, so I want you to remember these 4 factors. The last one is viral dose, meaning how many viral particles did you take in? Okay, so think about it. If you were just walking by someone on the street, okay, you might have inhaled a little bit of virus. Whereas if you are sitting next to someone who's sick on public transportation, like on a bus, and you're sitting next to them for an hour, okay, you're likely taking in a bigger viral dose. And that means your infection will end up being more severe. You have more viral particles that then can reproduce and spread. Okay, so the 4 factors again: how are you getting that infection? How healthy or strong is your immune system? What are the genetics of that infection? And how much of a dosage did you take in? Now this slide here, you have to know all of the examples for each of the organ systems. I am just kidding, okay? I'm mean, but I'm not that mean. So this is just to show you that there are a whole bunch of viruses that can infect you, and a lot of them are specific to different organ systems, okay? Some of them you've probably heard of, where others you may not have. And I do want to point out, now that it's the big thing, look, there's a coronavirus and it's under common cold. Okay, so that's just an example of how the media can go crazy with things. Now, when it comes to any of those infections, you just saw in the last slide, whole bunch of possible viral infections. When it comes to them infecting your body, they have to be able to spread through your body and avoid your immune defenses, okay? So when I say in that first bullet point that when these intracellular parasite viruses leave each host cell and enter the extracellular environment, it's dangerous for them. Well, think about it. Your immune system is going to be throwing a whole bunch of things at them, trying to destroy them. A lot of immune cells trying to engulf them, for instance. So viruses need a way to spread through your body, spread cell to cell, with, minimal risk to themselves. And this is one of my favorite slides because I think, for an organism that we say is not truly living, doesn't have a brain, these 3 ways that they avoid danger, pretty darn clever, okay? Very, very smart. The first way that they avoid your immune system or reduce risk to themselves is by infecting neighboring cells. They move to neighboring cells. That way, they minimize the amount of time that they're exposed to your immune defenses. Okay, so think about it. If you are walking from, the bathroom to the elevator in your building, or from one classroom to the classroom right next door, there's barely any dangers that can come to you. It's a very short trip. Whereas if you're commuting to your school from an hour and a half away, 2 hours away, you're taking parkways, highways, all kinds of bridges and whatnot, there's a lot of exposure time. There are a lot of different ways you can get injured along the way. So for them, going to neighboring cells reduces the risk of exposure, minimizes it. The second term is syncytia. What that means is they will form these giant masses by fusing many infected cells into one giant cell. So now they're able to move to faraway places without getting as much exposure. Okay, so with neighboring cells, they would only be, let's say, if they're infecting brain cells, they'll infect the next brain cell. With syncytia, now they can move all the way down to your feet if they wanted to, with minimal risk because they're hiding in your own clump of infected cells. So your immune system might not recognize them. The third one is my personal favorite. That's what the picture on this slide is. They release what's called decoy virions. Basically, they will mass produce a whole bunch of empty protein capsids, meaning empty virus shells, in large numbers just released throughout your body. Why is that a benefit to them? Well, if your immune system is so busy putting all of its resources trying to destroy these fake empty viruses that do not have any nucleic acid, can't do anything, then they're not necessarily going to catch the real viruses and attack them. Okay, so it's putting your immune system puts all of its resources trying to take down the fake guys, and it reduces the risk to the real guys. Okay, so make sure you're comfortable knowing the 3 different ways that a virus can kind of get by your immune system. Okay, neighboring cells, syncytia, and decoy viruses, and know exactly what those terms mean. Now, I want to spend a minute just going over some specific clinical examples of viral infections. Okay, so now the viruses has made its way in, it's cleverly bypassed your defenses, and it's infecting you. So many viruses end up hitting your respiratory system because think about it. You have to breathe. You are constantly breathing without even thinking, okay? So that is basically the most common portal of entry for these viruses to come in. Now, you have 2 parts of your respiratory system. You have your upper respiratory system and your lower respiratory system. The upper respiratory system is generally not as harmful of infections usually, whereas the lower respiratory system is going to be the more severe kind of serious infections. So now when you think of upper respiratory infections, think of the less problematic ones. The two most common upper viral respiratory infections are going to be the common cold and pharyngitis. Okay, common cold and pharyngitis. Whereas the lower respiratory, these will be more serious, and the most common ones for that will be the flu and pneumonia. And so now whenever you hear people talking about the cold and flu, and you kind of want to know the difference between them, well, think about it. Cold, common cold, is affecting your upper respiratory system. So usually if you get a cold, yes, you're uncomfortable, but you could usually make it around. A lot of people even still try to go to work or school, even though you technically shouldn't. You don't want to infect everybody. But you usually can if you have a common cold, meaning you're capable of, you're feeling good enough even though you're sick. Whereas when you have the flu, you are knocked out. You end up staying in bed. You usually have a fever and a lot stronger complications, okay, because that one is lower respiratory. Lower respiratory means your lungs are infected, okay? And your lower respiratory, such as your lungs, that's supposed to stay sterile. So when you have an infection there, it's a problem. So many viral infections cause what we call classic flu-like symptoms. So this includes things like fever, malaise, that yucky, drained feeling you get, anorexia, meaning you have no appetite, headaches, and a lot of body aches. Basically, a lot of these things are side effects due to your host's responses. And so when we say your host responses, it means a lot of these viruses will induce your immune system to release things like interferon and cytokines, which are meant to destroy viruses. But as they're destroying viruses, they're basically causing the result of immunopathogenesis. Okay, immunopathogenesis, which is things like arthritis or other inflammatory symptoms. Because, as you know, inflammation, fever, these are things your immune system does to try and get rid of pathogens, but they can also cause problems for yourself as well. Okay, so the release of interferon and cytokines causes a lot of inflammation and immunopathogenesis. Okay. Now to continue with the clinical examples, you can have a lot of gastrointestinal tract viruses. So the respiratory tract is the main portal of entry for infections because you're always breathing. But your GI tract is a common portal of entry as well because you're constantly eating and drinking as well. Now, when you see things like gastroenteritis, that's GI inflammation, and you end up with a lot of vomiting, diarrhea. But some infections you can get and not even show any symptoms at all. Now, one point I make on this slide is that a lot of GI tract infections that are viral tend to be naked capsid viruses, meaning that they don't have the extra envelope around them. They're just the regular protein shell. They're usually more stable, okay? But these issues, if you basically picture envelope viruses, that would mean that they have an extra lipid layer, and that can easily be neutralized by various chemical or physical agents. And basically the host cells would also be able to target them better. So by being naked capsids, they're a little more stable. But I want to point out that sometimes when you see these kind of infections and you see GI tract, you see things like vomiting and diarrhea listed. Most people don't take that seriously, but I want to point out that there are still a lot of third world countries where things like vomiting and diarrhea end up leading to a lot of death, especially in children and infants. There's a major loss of fluids and electrolytes that can ultimately be life-threatening, and there's also a lot of tissue damage. Now, the main way these things are spread are by fecal-oral spread, so always be extra careful about properly washing your hands and properly washing any foods, especially fruits and vegetables, that you're not going to be cooking. Okay, you want to get rid of any potential fecal-oral spread. In addition to the GI tract infections, you also end up with various viral skin infections. Okay, the three main ways of getting virus-induced skin diseases. One is from parenteral route entry. And what parenteral route entry means is any cut or any break in your skin. The second way that you end up with viral-induced skin diseases or problems is from what's called viremia. Okay, so viremia means that a viral infection of one of your other body systems has now spread or traveled. It's become systemic through your bloodstream, and then it can affect your skin because it's now throughout your whole body. And the third way a virus can affect your skin is through host inflammatory response. Okay, so what I was just saying in the previous slides, how your body tries to protect itself, but can end up hurting your tissues, and that includes your skin. Now, when we talk about skin infections, there's a few different terms that we use. So maculopapular. When you hear a macule or a papule, what that basically means is you have a blotch on your skin and it's raised up, but there's no fluid or anything in it. Okay, it's just a raised up lesion on your skin. Whereas vesicular, once you have a vesicular skin infection, that means that now it's a raised up lesion, so it's palpable, but it has fluid inside. Okay, so vesicular skin infections, the skin bumps will have fluid inside. Nodular is what we call a large papule. And again, remember, papules are palpable. They're raised up bumps on your skin, but they are not fluid filled. And then the last one is hemorrhagic. And hemorrhagic means that you have skin lesions that are bleeding. Okay, skin lesions that are bleeding. The last bullet point here shows that sometimes skin infections can cause a whole bunch of systemic symptoms. So exanthem is basically a widespread rash across your whole body, and it can include things like fever and whatnot. Hemorrhagic fever, now you know hemorrhagic, anytime you see that, there's massive bleeding involved. And then arthritis, you have inflammatory issues. Why would you have inflammatory issues? Well, again, your immune response. Okay, so I'm going to summarize. I know I said a lot on this slide and there were a lot of key terms. I'm going to summarize or have recap slide next for you to make sure you get all of the notes. Okay, so here is the recap so that you can write down the notes that I just said. Make sure you're very comfortable with this terminology, okay, because you know it's makes for very good questions on exams. Now you have viral diseases of organs and tissues. Okay, the two examples that I want you to remember of this are mumps and hepatitis. Now, whenever you hear mumps, I want you to know exactly which organ it's affecting. Okay, so mumps targets the salivary glands, which we call parotid glands. Okay, so mumps, which organ is it affecting? Your salivary glands, also known as parotid glands. Now, why is it not as common now? Well, that's thanks to the MMR vaccine, the measles, mumps, rubella vaccine. Then you also have hepatitis, which is another common viral infection of organs and tissues. Specifically, which organ is affected there? The liver. Okay, liver. And there are various ways that you can get different types of hepatitis. You can eat food that's been contaminated by someone who has the infection. You can get it from sexual transmission, or things like needle use, so drug use, or blood transfusions. Now, the next category here is the scariest, most problematic, the viral diseases of the central nervous system. Now, the big problem with the central nervous system getting infected is the idea of the blood-brain barrier, as well as your skull, okay? So what does the blood-brain barrier do? Basically, it's this network of capillaries, and it basically makes it so that most materials can't pass from the blood to the brain. This is a problem if you have an infection there in the central nervous system, because most drugs and antibiotics cannot make it to where the infection is because of the blood-brain barrier. The second problem is that infections tend to cause inflammation. Why is inflammation bad if it's your central nervous system? Well, think about it. You have a skull. There's no room for swelling in the skull or the spinal cord. That could be majorly disastrous, okay? So when you think of viral diseases of the central nervous system, know that they're very problematic. Most of them are lethal because of the idea of the blood-brain barrier blocking drugs or treatment, and because inflammation becomes so dangerous. You know, your brain can only swell so much before it starts squishing against your skull and getting damaged. So the last point that I want to make is that there are some infectious agents that are similar to viruses. So remember, whenever we say virus, you're thinking about an infectious agent that is just a nucleic acid, such as DNA or RNA, wrapped in a protein shell. The two types of infectious agents that are similar to viruses are viroids and prions. Viroids are simply a naked RNA infectious agent, meaning it is just nucleic acid with no protein shell. And this guy basically tends to infect plants. And so that's why, honestly, there's not much research on them, not much is known about them, because they're infecting plants rather than animal cells. But when you hear viroid, know that this is an infectious RNA, no protein. Prions, on the other hand, now instead of just being the infectious nucleic acid, instead prions are just the infectious protein, okay? No nucleic acid. Now prions, there's a lot more research getting done now because prions infect animal cells, including humans. They're responsible for very problematic neurological diseases such as Kuru and mad cow disease. Now, the thing with prions is, if you were to picture your own nervous system, your own neurons and central nervous system, you actually have prions, normal proteins that are part of your nervous system. But if you were to get infected with pathological prions, these are misfolded proteins. And the problems with these misfolded proteins is that when they're pathological and misfolded, they then go around infecting all the normal, healthy folded prions to misfold them. And if you think of proteins, proteins are defined by their structure, by their folding. If they're not folded properly, they're not going to function properly. Okay? So prions become very dangerous. And one of my students labeled me a prion long ago because she says, I go around corrupting all of these students. And that's what the prions do. They go around corrupting all of the healthy, good, normal prion proteins. Okay? So whenever you hear the term prion, there's a few things I want you to remember. First off, Prion is just protein, nothing else, just protein. Second thing I want you to remember is that you have normal, healthy prions. It's only a problem when they are pathogenic ones that start misfolding and misfold all the healthy ones. The last thing I want you to remember is that these misfolded proteins cause infections such as mad cow and kuru. And the problem with these infections is, as prion infections, there's no cure, there's no treatment. They can basically just make you comfortable. And usually within 12 to 14 months, you unfortunately die. And mad cow disease, you know, that's the one that you always see on the news and everything, especially with contaminated hamburger products. But the other one that I personally find fascinating is kuru. And kuru causes tremors and loss of coordination, neurodegenesis, eventually death, just like mad cow does. But what's fascinating about kuru was actually this is one of the first ways that they discovered infectious pathogens. And this is how, you know, the first area that we saw prions in. It was because of what I call nice, friendly cannibalism. Okay, so what kuru is that there was this tribe in New Guinea where they believed in ritualistic cannibalism. So if one of your loved ones died, well, you kind of always wanted a piece of them to be with you. So what they did was the women would basically prepare the body, you know, serve separate parts to different people. And it was specifically done in a way that, well, think about it, old-fashioned thinking, men have, you know, you want virility and strength. So men were given the muscle tissues, whereas women and children were given the brain to feed upon. So what they found in this tribe was only the women and children who were suddenly getting major neurological dysfunction and gradually dying, because they were the ones that were eating the neurological tissues that happened to be infected with prions. Okay, so whenever you hear kuru, I want you to think of cannibalisms showing us prion disease. And again, remember, there are no treatments, there's no cure for prion diseases such as mad cow and kuru. The patients end up unfortunately dying. Okay? On that sad and depressing note, that's the end of this lecture, and I want to see what kind of questions you come up with. So head on over to the discussion board, and for each student, make sure to write 2 questions that you would ask if you were coming up with an exam for this topic. Okay? If there was any part of this lecture where you thought I went too fast or you didn't quite understand it, or like I said, if you want any practice problems, for instance with viral titer, just send me a remind message and I'm more than happy to go over the material again with you one-on-one. Okay? Thank you and have a great day.